I'd like to take the liberty of moving towards the topic of genomics and personalized medicine, if we can even approach that topic. And how do you all feel that genomics can really revolutionize personalized healthcare? Or can it even do that? Can it approach that? So I don't know if anybody wants to take a leap first. Joe, I think.
Yeah, one of the things that's happening right now is that babies that are born with a condition that's unrecognized are immediately genome sequenced to identify what might be the cause. And about 50% of the time, the cause can be found from sequencing their genome. So I think already that's personalized, individual medicine that can be deployed. You know, what if there is a cure for that? What kind, if it's a metabolic disease? I think that the challenge, you know, is that it's kind of expensive still, and that thinking about the kind of inequities that could be caused. Most of the databases that are filled with sequences are mostly Eastern Europeans, and so or Europeans in general. And so I think there's an equity issue that's involved with genomics.
How is that being handled that's a US perspective, is that similar in Japan?
I see. At this moment, it practically not so much, going. But in the research level, we had a many-year program, so sequence many different type of cancer patient. So then I think from that, big data come out, and then people try to personalize the medicine or the methods. So then in future, coming soon for more social the approach, yeah.
Well, Joe gave us an example of diagnostics. I guess it's genetic diagnostics. What do you think some of the most promising developments in gene therapies are related to humans in this respect? I don't know, Andy or Rich?
I mean, there is better and better delivery, but it's not perfect at the moment. So there's quite good delivery to
So you mean delivery, so clarify that a little bit more.
Well, if you want to do gene therapy, you have to put a gene into a cell, and that means that something has to go across a membrane. And some cells, that's relatively straightforward, and some it's quite difficult. And so liver is great. So people have found ways to deliver things to a high fraction of liver cells. It starts to get worse after that.
So liver, lung, pretty easily accessible in that respect, but maybe muscle tissue or?
Kind of okay if you want some of the muscle, if some is okay. Brain is going to be harder, and these things are all, I think, being worked on, and there are great advances in them, but it's going to be slow, and there'll be time required for each stage of the process to understand what the downsides are to this kind of access.
Rich, do we know enough to be able to do this in humans, targeted gene therapy?
I think we know enough to be careful. And I think this is the most important thing. But there are plenty of people working on different approaches to getting genes into cells, getting messenger RNA into cells, and so on. I mean, if you see what happened during the COVID epidemic and the vaccines that were produced, these were produced not because that was where the research was heading. The research was heading to treating disease using messenger RNAs, and it got adapted for that. And I think this is something we're going to be able to do in the not too distant future. I don't think it's going to take too long. A lot of people are working on it. But of course, in the US, unless the administration changes its policy towards scientific research, we're not going to make a lot of progress.
That in itself is a challenge, right? But what are other challenges that we face as scientists or ethicists as we embody using genomic medicine, I guess you'd call it?
One of the major challenges is this thing that when people have a new idea that involves a therapeutic, the whole infrastructure of pharmaceutical companies is very excited. If there's a new idea that's a diagnostic, it goes nowhere. Everyone says, no, you can't. So the sort of commercial infrastructure says, no, you can't sell a diagnostic. Nobody's going to pay for it. And the other part of it is if you give a complicated molecular diagnostic to physicians who aren't trained to use it, they don't know what to do with it, actually. And so there's an AI problem perhaps there, or some kind of I problem, is that the more it's a black box, the more difficult it is. And so it's almost a reverse AI thing, as if the more we can have physicians really trained in how to interpret genomic data and work with it, and the more we can take that data and provide information that's actionable to physicians, the better off we are. And at the moment, there are probably quite good diagnostic approaches that could be there that just aren't happening because the industry is saying, oh, we don't want to deal with that. Give us a therapeutic. Give us a drug you can give to somebody and make them better that we can charge them for, because the economics are hard for drugs.
Except they don't want to make you better. They want to give you something you've got to take for the rest of your life.
Yeah.
That's where the economic argument is.
Right.
Yeah, in Japan, I think, there's so much, the carefully thinking about that. So even though we developed, the methods like a drug delivery system or using, join the chemistry people also, but maybe if the other country start and permit it in a social acceptance and going, that time, then we will follow.
It's easier to adapt.
Yeah, yeah.
So, all right, there's challenges in terms of understanding the genome and recognizing that a lot of that information comes from perhaps one region of the world. So we need to think about just the globe right now. There's probably challenges in terms of developing what to target, still thinking a little bit about those questions, and then delivering these medicines in a way that is equitable. I guess those are some of the big things that we're looking at. Now, I do want to turn to the audience, and I hope we get some good questions from all of you, but we have microphones, so if you are someone who wants to ask a question, kind of stand up, and we will get a microphone to you. And the questions we're discussing right now is, how can genomics revolutionize personalized healthcare? That's the first topic we want to take here. Quiet audience. Go ahead, stand up. And it's always good if you tell us who you are and where you come from. And thank you for being the person who asks the first question.
Thank you for the opportunity to ask you guys a question. I'm Asako Kitai from Tokyo Medical School. I'm a fourth year medical student. One thing I wanted to ask was except cancer, what kind of I think cancer will be the main topic about regarding the genomics and application of genomics for this therapeutic reason and diagnostic reason. But other than cancer, what kind of disease are you guys interested in to apply those type of?
So the question, I think, really, where is the opportunity for treatment? And you say cancer is one of the obvious ones, but what other diagnoses or which other disease types can one imagine?
Well, there's already a therapy for sickle cell. There are two different approaches to be able to monitor activation of a fetal hemoglobin and also editing of existing mutations in hemoglobin. So there is already, and that's of course easily accessible because it's blood. But I think that already is telling us that that the delivery methods, if they come, if we can put them in the right place at the right time, it's going to be a bright future, I think, for personalized medicine.
Right. The problem there is the cost is so high that it can't get to most of the people who need it. And I think we have to find ways to lower the costs for treatment like that.
Health equity, health disparity is a real thing we have to wrestle with.
Yeah.
Go ahead.
Different approach. For example, everybody know the gut microbes and the humans relationship. So some researcher working on how to change the gut microbes. Sometimes you can use like a phage therapy or the other, or how to maintain the good useful microbes. And sometimes maybe you're engineered or in future. So not only your cancer directly, we had but many methods to indirectly control our body using a microbes also. Yeah.
Okay. Are you happy with those answers? Pretty good.
Can I ask one more question? Regarding genomics, I think epigenome will be very important, especially when you're targeting the disease for adults or elderly. And in that sense, like, what do you think, what is a limitation, and what kind of technology will be needed to to overcome those limitations?
All right, so you're really interested in the field of epigenetics or epigenomics, where you have a genetic code, but the environment that you experience might modify the way in which the DNA recognizes other factors and turns on or turns off genes. And that would be the epigenetics that you're interested in. And I guess your question's related to how can you take maybe the environment into account at the same time you have some underlying genetic difference that you want to treat. I think, is that sort of your question?
Yeah, thank
All
you.
right, maybe Joe, I'll start with you.
Well, one thing is we're still very early in trying to understand what the epigenome is of individual cells. That's some area that we've been working on with colleagues around the world as part of the BRAIN initiative. I think until you understand the role of epigenetics in a particular disease, there's nothing to treat. And so there are a few diseases or tools that you could develop that would silence a gene without inactivating it, without mutating it, but permanently silence it. And that would be there are several companies that now are building epigenetic tools that can be added to viruses, for example. So instead of taking a virus and inactivating a gene, you could activate it, for example, or you could silence it temporarily. And so there are there are companies that are focusing on epigenetic therapies, and I think it's still very early days because we don't know a lot about how much somatic variation there is in our body in terms of epigenetic variation. We're still because this is a cellular phenotype, right? Each cell in your brain, I would predict, has a different epigenome, and until you understand enough about that, it's going to be hard to compare. So, for example, diseased brain, we do this, compare Alzheimer's versus non-Alzheimer's, but most of the time the samples that you get are from people that already have damaging brain disease, and it's impossible to tell. I think that there are there are studies that are needed to assay individuals earlier in the course of the disease in these prospective kinds of experiments, and then you'll be able to identify what you could potentially do if there is a phenotype that's observed in a cell.
Go ahead, Andy.
There's another dimension to all of this, which is that in addition to cancer being a major target, a lot of us, and infection, a lot of us get sick because of autoimmune disease. And there's a genomic component to that, which is very interesting and intricate, which is that we have all the different rearranged immunoglobulin genes and T-cell receptor genes, two sets of genes that actually physically rearrange in blood and bone marrow. And those rearrangements actually determine at any given time a part of what our immune system is reacting against, either for good or bad. And so understanding those profiles will be quite valuable, and eventually being able to intervene either through direct mechanisms targeting those immunoglobulins or through playing with the innate immune components that actually determine the volume of the signal that comes on. Those will be quite valuable, and I think a very interesting future for genomic medicine.
Yeah, I would add a little to that. The epigenome is unbelievably complicated, and we understand virtually nothing about it. Good area for students to work in. I think there are some very valuable experiments can be done, some good studies can be done. When it comes to looking at genomics and personalized medicine and so on, one of the things that is easy to think about and deal with is when it's monogenic, when it's a single gene that is causing the problem. Not the case most of the time. Most of the time there are multiple genes. We don't know well how they interact. Another great area for students to get involved in. I think there are going to be some big discoveries to be made there, so there's something to work on.
All right, thank you for the questions. There's others, go ahead. And we have a gentleman here. And you're going to have to stand up if you have questions so that we can see you a lot easier. There's some questions in the back, so. Kind of got everybody warmed up.
My name
with
is
that first
Young
question.
Nam from USA. I'm sorry for young folks instead of old men. Well, I have a question about genome technology can eliminate congenital defect, like a birth defect. For example, epilepsy or Down syndrome. That's my question.
That's good. I mean, so there are certainly a lot of activities in the research field, probably things like CRISPR or microRNAs, RNAi technologies. Do you think that this is a reality going forward? Genetics for congenital?
I would say absolutely. It's just that the technology is not completely perfect at this point. I think it's improving quite a bit. And I think for devastating diseases, it can be deployed, I think, without a lot of ethical concerns. But mucking around with other genes that are not related to any major disease phenotype, I think, has to be avoided.
So very specific effects rather than...
Life-threatening diseases that could be edited out efficiently, it's still early days, but I think the tools will obviously improve.
Okay. All right, great. Go ahead in the front here. Good if you can remind us who you are and where you're from.
Thank you very much. I'm Zeynep from Osaka University, Immunology Frontier Research Center. I'm an assistant professor there. I have two questions. One of them is maybe not very related with us or you, but like I just want to have your opinion about, let's say everything goes really well and we have these technology really applicable on humans, but as you said, it is going to be expensive. What is the social effects of this? Like how can we overcome this as scientists? Maybe it is not our problem because we are just researching, but I don't believe that the scientists just only work as like researching. We have to think about the social problems also. So the richer will get the treatment and the poorer will die by themselves alone. And the second question is like, let's say these...
Let's take the first question
okay.
first, okay?
All right.
So, because that's an important question. And who's going to bear the cost? And how do we make medicines that are affordable and accessible to the people most in need? Any thoughts there? Go ahead.
So I don't know if you've heard of the Serum Institute in India, but this is a group that was set up by a very wealthy Indian, Cyrus Poonawalla, and it was set up to deliver vaccines, to deliver medicines to poor people. It was deliberately set up to do this. They came up with an early vaccine, made it available around the world in huge quantities. They are actually tackling sickle cell disease. They want to make that a very cheap treatment. But the problem always comes up with competition from the big pharma companies. They're interested only in money. They couldn't give a shit about anyone else, to be honest. They do not like cures. They don't like anything that cures a disease. This is why antibiotics are not on their radar so highly. And I think we have to get very careful about this, but we have to speak up and we have to encourage more institutions like the Serum Institute in India to get involved in making sure that these treatments and cures and whatever we can do are available cheaply without trying to make massive amounts of money from it.
There's another company that I know of that was spun out from a pharmaceutical company that does N of 1 research. So if you have a particular disease, rare disease, they will design for you for no cost an antisense oligo that targets that gene, and there's no cost to the patient. So, but it can't be something common because that would compete with the pharmaceutical industry. But they'll tailor it to. So there are those sort of efforts, but it needs to be expanded. It's as Rich said, it should be on the radar of pharma, but it's not.
I mean, there's a really major concern here, and a benefit for a type of science we don't have represented here, which is economics. You know, the health is a public good. It's better for everybody if people are healthy, and that means that some research and some treatments have to come from the public funds, either nationally or internationally, and there's benefit from that. How do we do that economically is something that, and commercially is something that has to be looked at. And there are models for, as Rich mentioned, there are models for companies that do work toward public benefit. And how do we scale those to deal with the medical problems of the world? Really important question. I would put it up in there in the same parallel as how do you get a gene into a brain cell?
Well, because you're right. If you have the innovation, but if you can't use it because it's cost prohibitive, it's really already then you've lost a great opportunity. So we need to get all those parts of the puzzle in the right place.
Right, but government regulations also screw a lot of innovation because they say, oh, you've got to spend huge amounts of money to bring it to market. That's not good. That's something that should change too.
Right, the amount of proof that you have to present to be able to get drugs approved, now the bar is very, very high. That's for certain. You did have a second question, and then I want to move to another individual, so go ahead.
Okay, the second question is a bit maybe strange again, but like, I just wonder, let's again get the best scenario. Everything is applicable, so we are able to treat any kind of disease, but I mean, until when? Because like if we treat every disease in a good way, and if we, for example, stop every genetic disease, so we are going for the perfect human, undying. So, and the people are not, like humans are not really good, that we saw this in the history. So this ideal human being may return into a really bad situation again. So who is going to be the decision maker in this situation, that until which step the genome editing is applicable for which kind of diseases and what kind of like people?
All right, so that's a great question to consider. If we have it within ourselves as scientists to be able to innovate, to have therapeutics for everything, and we can create a situation where people could essentially live, I don't know, maybe she's speculating indefinitely. Who's going to take the decisions about that and the extent to which people can remain on this planet? I guess that's maybe what you're trying to go for in that question.
Great.
But go ahead.
Thank you. Actually, the final decision is yourself. Yeah, because once law fixed, and if everybody can access that times, finally, who decide to use that technology? Maybe there is a many card you can choose. I think finally we have to decide by yourself. Yeah, it's my just opinion. Yeah.
Okay.
Right.
Rich, go ahead.
Yeah, the problem with that is there are lots of people who think they want to live forever. And so they're going to make the decision and say, hey, I'm going to get everything done.
I see.
You can't leave it to the individual, I'm sorry.
Yeah. So maybe you mean the upper limit, right?
Well, to some extent, there is a bit of that today because, we do have choices about whether we accept a particular treatment or not, to some extent. But you're saying there's a risk that there'll be people that will just take everything.
We know such people exist. They formed a society, the longevity society. They want to live forever.
I think they're called cronies. All right. So, and Andy, did you want to contribute to this?
Yeah, I mean, it's an interesting, when in the early stages of my career, it's a slightly different question, but graduate training consisted of often asking the students, if you had infinite resources and could address the problem of human longevity, what would you do? And I actually brought up a question to a very wealthy person at one point who I said, you really should support our graduate program. And what they said was pretty wise, is no, I won't support your graduate program because you train people to be in the most high-resourced environments and to be doing research towards questions that are focused on those environments with infinite resources, which is what we did. And so I thought that was very smart. And after that, I would ask students more question of, you have limited resources. You can't solve all problems. You don't have all resources. How would you use them? How would you use not $1 million, but $30,000, which sounds like a lot, but it's not much in research? And that's a really good question to ask people, and especially really idealistic young people, because they may come up with answers that are not, let's make people live a week longer, but let's deal with childhood disease. Let's deal with, the burden of insect-borne scourges in the world, all sorts of things that we could deal with probably more cost-effectively if we were thinking about it from that perspective. And that also enables people in all the world, in different places, to begin having the tools to be able to deal with problems in their community, which I think is going to be really valuable. And so I really, given also what's going on in the US, I really strongly feel that the advances in medicine are not going to necessarily come from the US and maybe not some of the developed world. And so there is an opportunity for countries that maybe don't have the resources, but have the desire and have the brains, absolutely, to be able to make really substantial advances.
So sometimes being forced to prioritize may really filter away not the nonsense, but the things that maybe are less acute to help you pinpoint the things that really need to be addressed.
Right.
All right, we had a question way in the back. Someone was having their hand up. Yep, well, there's two people there. So the gentleman, you can, and then we can take the woman just to your left. And again, who are you and where are you from?
Thank you. I'm Shudipal. I'm a postdoctoral fellow in Chiba University, Japan. So my question is, researcher now have developed some experiments to find out that epigenetic dysregulation is one of the hallmark of aging. So although mice models and in vitro cultures from premature age related disease patient-derived cells, they uncovered that epigenetic dysregulation is the hallmark of aging. So my question is there in the future, is it possible to incorporate genomics in humans, actual humans, to slow down the aging process or to prevent the aging-associated diseases, to promote healthier aging in population? Thank you.
All right, good question. Lots of interest in epigenetics from this group here. They care a lot about maybe the epigenetic drivers, how the environment is influencing our, in this case, longevity, on top of the genetic code we inherit.
Rich said this was a good area for students to go in, and I agree with him. And that's because we still need to learn a lot. I mean, thinking about altering the epigenome in what cell would you change for aging? You know, would you work on the brain? The heart ages at a different rate than another organ. So I think there are a number of companies that have funded by billionaires that want to live forever, and they want to use the latest technologies to try to reverse the aging of a cell, which exists. You can make from the work of Shinya Yamanaka, and you can take a cell all the way back to an embryonic state. But you don't want to go back that far because then you get a tumor. But you can maybe go back part way, and there are a number of companies that are doing that, trying to reverse aging in mouse models of aging, for example. So I think it's a long ways away. I think for first finding out what yes, there's degradation of the epigenome with age. Many labs have reported that, but exactly what you would do to change that isn't really clear. We need a lot more bright people working in that area.
And I think what Joe's saying, there's also an uncertainty. If you took a person who, without intervention, would get older and sick and uncomfortable and have the epigenetic signature of aging, and you got rid of all the epigenetics, would you get someone who lives forever, or would you get someone who got older and sicker and uncomfortable but didn't have that signature? And I don't think we know the answer to that question yet. There definitely are correlates of aging that are epigenetic, and probably you would have some different conditions that would be better off. But I'm not sure exactly what you'd fix with that. It's a good experiment. I'd love someone to do it. Tell me when it's done.
Sometimes I think it's better to let nature take its course. I did a epigenetic test for biological age and what have you, real age. I'm 16 years younger biologically than I am chronologically. I was very happy with that. I don't want to live forever. I'm happy to die when the time comes.
But you're happy to be 16 years younger than you. Yeah, okay. Did you want to add to this?
Yeah, that is really important, the research. So everybody mentioned about, that is useful to work on that. But I think it individually had a different, opinion to long, the aging, the stuff. So then there's just a marker, as you said, if you're younger, it's fine, but it doesn't matter if you're longer, right? So it's a depend on the personality and then the opinion into a life, yeah.
Right. Personally, I think the thing that kills more people than anything else is worry, stress and worry. Don't worry, you will live a good long life.
All right, there was a woman that was just to your, that hand her the microphone, and again, thank you for your question.
Hi, I'm Anna from Israel. I'm a medical doctor currently doing my PhD. And my question is, what do you think the role of genomics would be in prevention?
Okay, so we talked about it, I guess, in diagnosis, but this is prevention. So if you knew you had the gene for, I guess, diabetes, what would you do?
Take insulin?
Yeah.
Oh, sorry.
Yeah. Preventive medicine? I think it's
I
one of
mean,
the
if you
hopes,
could-
isn't it?
if
Isn't
you-
it one of the hopes?
If you could take enough genomes and enough patient data to predict outcomes at an early stage, you could say, well, you've got, I mean, this is totally separate from environment. That's another factor. But if you captured all the information that you could about the environment and then said, okay, with this genotype, you're more susceptible in this environment, eating McDonald's hamburgers, whatever, that you will likely get diabetes. Okay, so start now to change your diet. So change behavior rather than you're not going to change the person's genome at that point, right? Unless they have a single Mendelian gene that you could, and even then that would be challenging. Even cystic fibrosis, which was one of the first genes identified, still to this day, there's no gene therapy approach yet. I mean, it's not in routine practice.
Right, so a lot of it is risk. You look at your risk for different gene diseases, not always necessarily meaning that if you have a increased risk of diabetes genetically, I'll use that as an example, it doesn't necessarily mean you'll get diabetes, but there are some where you are certain to get disease. Okay, other questions? We have time for just a couple more here. Okay, over here. You're gonna have to run with those microphones. There we go. And I think we had 2 hands up here and we had 1 hand over here, and I think that's all we'll probably have time for, just so people are prepared for that. So short questions, okay?
I'm Ryo Shingubara, biogeochemist from National Agriculture and Food Research Organization, Japan. But I'd like to ask a question as a mental disease patient. I'd like to ask if the genetics can have any potentials in the field of mental disease or the disease of mind. This is my question.
All right, so what's the evidence
Some
that
of your
there's a
perspectives.
genetic component to psychiatric disorders or mental fitness?
Perhaps one possibility is to understand the mechanism. For example, depression disorder is not understood fully yet. Only 60 to 70% of the patients can be better with serotonin-related medicine, SSRI, but the other part is not unknown. And yeah, so I mean, the brain is very different from person to person, so I wondered if it can be related with.
Right, great, thank you. We
yeah.
have to have short questions here, so go ahead.
One of the approach. I mean, so I don't know that genomics will solve any of those questions instantly, or genomic intervention. I mean, the best that can be said is that genomics can often look at cases where there's a very clear single gene component that's involved in a disease, and at that point identify the gene and then study that gene with all the tools of medicine and biology and physiology to understand what's going on, and then design an intervention that may then be useful beyond that individual rare disease family that has a mutation. And so that's one approach to that. But it's a long road approach, and we need to know more about how the brain works to be able to begin rationally intervening. And as you said, that the current medications, although they can be really, really valuable for some fraction of people, they're very, very rough tools, and there could be better tools out there. And we'd hope that research all over the place, including genomics as one, maybe small part, could really benefit that.
All right, great, thanks. I'm going to have to move on. Go ahead. Now, it has to be a short question, okay?
Thank you. My name is Gong Lin Xiang, and I come from Kanazawa University Cancer Research Institute. And I have a question here that now we have lots of mRNA vaccine. As we all know that mRNA vaccine are widely used during COVID-19, and also last month a paper come in from Nature also said mRNA vaccine can completely cure the pancreatic cancer. So, but mRNA vaccine still remains some problems. It can cause some known and unknown problems in our human body. So my question is, do you still believe this mRNA vaccine is a good way to cure some disease?
I mean, it's a wonderful approach to have in our quiver of things. It won't be a cure-all for everything. And you know, curing, I don't know the paper you're describing, but it might be a miracle, and that would be great. Sometimes the best of studies in a small scale are harder to scale up for one reason or another. I look forward to mRNA vaccines being one of the tools that we use. We'll get to know more of it. Clearly, you know, we've done okay with it. The health of the world has not seriously been hurt by the COVID-19 vaccines, and I'm thankful for that. I'm thankful that they've saved a lot of people. You know, the next stages in that world, we don't know. There'll be some great applications, and there'll probably be some challenges that come up.
All right, that's great. We had one more question. There was one more over here. Last question, yep. Again, make it short, because we don't have a lot of time left.
Thank you. My name is Masaki from University of Tokyo, another medical student from University of Tokyo. My question is ethical aspect of genomics. You know, people having cancer genes might have to pay more price for insurance, or people can have like prejudice against having baby if you have cancer genes or something like that. So what do you think is the ethical problem around the genomics, as well as what would be the roles of scientists to play?
All right, that's a good question, and that'll be the last question we have. So just ending on the ethical responsibility, the ethical aspects of this. Does anybody want to take that on?
Well, right now there are in the U.S. laws against discrimination by insurance companies, for example, based on genetic data. It's not bulletproof, but there's every attempt to keep the information private in a lot of the projects that are, for example, the All of Us project, which is to sequence a diverse population of individuals, more diverse than in the past across the U.S., and that data can be accessed under very strict conditions so that there can't be discrimination of that family. And often it can happen when people put their own data out there, and then, you know, maybe because they have relatives that, you know, that is, you know, under some circumstances, then you can say, oh, maybe you're going to get this disease. So I think people should be a little bit cautious about release of their genetic data at this point, for fear that maybe in your country the laws are not as strict as in others.
All right, I think we're going to have to end here, but this was really fantastic. We've gotten a lot of insight in terms of where genomics can take us, a lot of interest in diagnosis, also for potential treatment. With that also, we need to think about the impact on society and making certain that these medicines can be deployed to all the people in need. So I want to thank the panel again, and all of you in the audience who participated for your energy today. So thank you.
Thank you.